Hello everyone, I am Dr. Renu Kohli, Associate Professor in Geology at Government Bangalore College, Pali. Today we will talk about antigen. They are also called immunogens. What is antigenicity? What are paratops and epitopes? Types of antigen on the basis of their origin. Types of antigen on the basis of antigenicity. What are antigens? Antigen is a substance that is capable of stimulating an immune response, usually protein in nature and sometimes polysaccharide, that generates a specific immune response and induces the formation of specific antibody or specialized sensitized T cells or both. An antigen may be a substance from the environment such as bacteria, virus, cancer cells, and other foreign substances like pollen, or it may also be from inside the body. The antigens are mostly conjugated proteins like lipoprotein, glycoprotein, and nucleoprotein. Since antigens are associated with immune system, they are also called immunogens. Now, what is antigenicity? Antigenicity or antigen reactivity is the capability to induce an immune response. It refers to describe both the ability to elicit immune response, that is to induce the production of antibodies, and capability to react with antibodies. For example, when B cells get activated, it secretes antibodies, and the antigen which have property of antigenicity will combine to these antibodies. Now, what are paratops and epitopes? A paratop. A paratop, also called antigenic binding site, is a part of antibody which recognizes and binds to an antigen. The part of antigen to which paratop bind is called epitope. Epitope is also called antigenic determinant, is part of the antigen that is recognized by the immune system, especially by the antibodies. B cells or T cells. For example, the epitope is the specific piece of antigen to which an antibody binds. Means epitope are part of antigen and paratopes are part of antibodies. Types of antigens on the basis of their origin or on the basis of source and immune response. There are three types: exogenous, endogenous, and autoantigens. Exogenous antigens are those antigens which have entered into the body from outside, for example, through inhalation or ingestion. Exogenous antigens are the most common kind of antigen, and they include pollen or food that may cause allergies, such as molecular compounds of bacteria and other pathogens that could cause an infection. These antigens enters the body or system and start circulating in the body fluid and are trapped by APCs or antigen processing cells, such as macrophages, dendritic cells, etc. The uptake of these exogenous antigens by APCs are mainly mediated by phagocytosis. Example, bacteria, fungi, etc. What are endogenous antigen? These are the antigen which originate from own body of host organism. These are body's own cells or subfragments or compound or the antigenic product that are produced. The endogenous antigens are processed by macrophages, which are later accepted by cytotoxic T cells. Example, blood group antigen. Now, what are autoantigens? An autoantigen is usually a normal protein or complex of protein and sometimes even DNA or RNA that is recognized by the immune system of patient suffering from a specific autoimmune disease. These antigens should not be under normal condition the target of immune system, but due to mainly genetic or environmental factors, the normal immunogenic tolerance for such an antigen has been lost in many patients. Example, nucleoprotein, nucleic acid, etc. Types of antigens on the basis of antigenicity. They are two types: complete antigen and incomplete antigen, which are also called haptens. Complete antigens are those antigens which generate immune response by themselves. They are generally molecule with high molecular weight, that is molecular weight more than 10,000 daltons. Incomplete antigen or haptens are the substances which have low molecular weight, that is less than 10,000 daltons, and they are itself not capable of stimulating antibodies formation when administered by themselves unless it is coupled to a carrier molecule. A hapten is essentially an incomplete antigen. For example, the molecular weight of penicillin is only 350 daltons. When injected or ingested, penicillin reacts chemically with protein in the body to form hapten carrier complex, which incite the production of antibodies. The formation of hapten carrier molecule causes the hapten to become immunogenic, resulting in the production of anti-hapten antibodies. Upon second exposure, these antibodies can recognize and bind to the same hapten, even in the absence of carrier molecule. Therefore, haptens need a carrier molecule to become immunogenic. Once they become immunogenic, they are also antigenic, and they are able to bind to antibodies or other component of immune system primed by the hapten carrier molecule complex. So the haptens have two characteristics: a substance that is non-immunogenic and a substance that has the property of antigenicity but not the immunogenicity. Thank you.